Hi, I'm Leah Kent from StemGen. Today we're going to show you how to thaw and freeze human embryonic stem cells. So let's get started.
To thaw human embryonic stem cells, a cryovial is removed from liquid nitrogen and quickly thawed in a 37 degrees Celsius water bath. The cell suspension is transferred from the cryovial to a conical tube, diluted with human ES cell culture medium, and centrifuged to form a small cell pellet. The supernatant is then removed, and the cell pellet is resuspended with fresh human ES cell culture medium in the tube before plating on one well of a 6-well cell culture plate previously plated with an irradiated MEF feeder layer. One day prior to thawing human ES cells, plate a feeder layer of irradiated mouse embryonic fibroblasts, or MEFs, in MEF culture medium in one well of a gelatin-coated 6-well tissue culture plate. Incubate the plate overnight at 37 degrees Celsius and 5% carbon dioxide. To ensure an efficient and successful thaw, be sure to have all necessary equipment and reagents ready before removing the human ES cells from liquid nitrogen. Using metal forceps, remove a cryogenic vial of human ES cells from the liquid nitrogen storage tank. Roll the vial between gloved hands for 3 to 5 seconds to remove the frost. Record the information on the label of the vial. Using metal forceps, immerse the vial into a 37 degrees Celsius water bath. Swirl the vial gently and observe the progress of the thaw often but quickly by holding the vial up to the light to see the size of the ice crystal. Do not submerge the cap of the vial in the water bath, as this could contaminate the cells. When only a small ice crystal remains, submerge the vial in ethanol to sterilize it. In a sterile biological safety cabinet, transfer the contents of the cryogenic vial directly to the bottom of a 15 milliliter conical tube. Slowly add 4 milliliters of human ES cell culture medium to the tube. Gently rock the tube to continually mix the cells as the new medium is added to the tube. Centrifuge the human ES cells for 5 minutes at 200 Gs. While the cells are in the centrifuge, retrieve the previously prepared MEF feeder plate from the 37 degrees Celsius incubator and label it with the appropriate human ES cell information, such as cell line, passage number, and thaw date. Aspirate the MEF culture medium and add 1 milliliter of PBS to the well. Bring the pelleted human ES cells back to the biological safety cabinet and carefully aspirate the supernatant. Be careful not to aspirate the cell pellet, but remove as much supernatant as possible, as this solution contains DMSO from the freezing medium. Resuspend the pellet very gently by adding 2.5 milliliters of human ES cell culture medium using a 5 milliliter glass pipette and pipetting 3 to 4 times. Aspirate the PBS from the MEF feeder well and slowly add all 2.5 milliliters of the human ES cell suspension to the prepared well of the 6-well plate. Place the plate into the 37 degrees Celsius incubator and carefully slide the plate forward to back and side to side to evenly distribute the cells throughout the well. To avoid concentrating the colonies in the center, do not swirl the plate in a circular pattern. Allow the cells to attach at 37 degrees Celsius and 5% carbon dioxide overnight. On the day following the thaw, remove the medium with any floating cells and add 2.5 milliliters of fresh human ES cell culture medium to the well. Incubate the plates at 37 degrees Celsius and 5% carbon dioxide overnight and continue to culture until the colonies are ready to be passaged. human ES cell freezing protocol begins with healthy, medium-sized human ES cell colonies cultured on a 6-well plate. After incubating the culture with collagenase 4 enzyme, the colonies are scraped with a 5 milliliter glass pipette, collected in a conical tube, and centrifuged to form a loose cell pellet. After aspirating the supernatant, the cell pellet is resuspended first with human ES cell culture medium and then diluted with 2x cryopreservation medium. The cells are evenly distributed in 1 milliliter aliquots and frozen in cryogenic vials. While the cells are in the centrifuge, label the cryogenic vials inside the biological safety cabinet with the cell line, passage number, and freezing date. The typical density for freezing human ES cells is 1 well of cells from a 6-well plate per cryogenic vial. When the human ES cells have been centrifuged, bring the tube back to the biological safety cabinet. Aspirate the supernatant from the tube, being careful not to disturb the loosely packed cell pellet. Resuspend the cell pellet with the appropriate amount of human ES cell culture medium, 0.5 milliliters of human ES cell culture medium per cryogenic vial. Be very gentle when pipetting, as the human ES cells recover better when frozen in relatively large colony pieces. Slowly, while gently shaking the tube, add the appropriate amount of 2x human ES cell freezing medium to the cells. Once the freezing medium is added, mix by pipetting the solution extremely gently 1 to 2 times. Quickly but gently, add 1 milliliter of the cell suspension to each cryogenic vial. Transfer the vials into an isopropanol freezing container. And place in a -80°C freezer overnight. The cells will freeze at 1°C per minute in the isopropanol freezing container. On the following day, quickly transfer the frozen vials to a liquid nitrogen storage tank using metal forceps. The first day after human ES cells are thawed, the small colonies may appear transparent and can be difficult to see under the microscope. Since newly thawed human ES cells tend to proliferate rather slowly, they may take a few days in culture to appear as established colonies.
We've just shown you how to thaw and freeze human embryonic stem cell cultures. So that's it. Thanks for watching and good luck with your experiments.